just like to welcome you all here at the Old Mouton Hall, University of Louisiana, Lafayette. I'm Ray Grasser. I am the coordinator of the anthropology program in this department. And it just tickles us a great deal to collaborate with the people that are here today and to welcome you all to this event, which is very important for science and for humanity, of course, as well. We have an anthropology society, a student group that's helping to host this event too. Mr. Ramsey Purple is the president of that group, and we're glad he's here with us today. And of course, we have a lot of very important guests and speakers here that we need to learn from. But I just want to say welcome, and if there's something I can do, please let me know. I'll be around here and looking forward to hearing about the program. Of course, the Sierra Club is a major host for this event as well. And we have Mr. Harold Schefter with us today. I'd like to invite him to say a word.
I just want to say thank you all for being here. I want to especially thank the presenters who are here to educate us about a very important subject to us all. And I want to especially thank Dr. Ray for hosting this and putting this together. Thank you.
All right.
Emily? I'm Haywood Martin. I'm vice chair of the Acadian Group Sierra Club here and chair of the statewide Sierra Club. And we're happy to be involved in organizing this event. Thanking Ray and thanking Emily for coming together and all the folks that are helping on this presentation to put it together. So Emily Long Douglas with Sea Grant was the real person that initiated this. She came to one of our meetings about 8 or 9 months ago and said we'd like to do something. And we came up with a plan to send her a list of things we'd like to see, and one of them was about this person. So here we are. This is really great that this came together. So Emily Long Douglas with Sea Grant.
Thank you so much. And also thank you to the Sierra Club and to everyone at ULL, the Anthropology Society. It's really great that we could have this seminar at this venue. So I'm Emily Mong Douglas. I'm from Louisiana Sea Grant, and I'm also representing the Gulf of Mexico Sea Grant.
We're working on it. Thank you.
All right, so we're working out some technical issues maybe. But so I'm with the Louisiana Sea Grant and the Gulf of Mexico Sea Grant Oil Spill Outreach Program. And I'd like to let you know first that we'll be having the seminar itself. I'll be talking a little bit about our oil spill outreach program, some neat resources that you all might be able to go home and use this evening even, and maybe set up a little bit of framework for what our guest speakers will be talking about. Drs. Andrew Nyman and Chris Green from LSU Ag Center. And lastly, after that, we're going to have a question and answer session. We're going to save the questions for last. And then just after that, I hope that you'll all stick around and fill out your input and evaluation forms. They're in these folders that are out front, and also pens are available as well. And so inside there, you've got a one-pager about our program, 2 brochures that Drs. Green and Nyman and their grad student, Bran Wagner, have come up with to help people understand oil and toxicity. And then this pocket here has the input and evaluation form. And we're really using that input and evaluation form to understand what things you really care about and what questions we've answered for you and what questions you still have, so that we can create more seminars like this that are better targeted to your needs and bulletins that suit your interests. So to get into it, what, this? All right. Okay. All right, we'll figure this out. We'll figure it out together. Okay, so as I mentioned, I'm with the Sea Grant program, the Gulf of Mexico Oil Spill Outreach, and we've been sponsored by GOMRI, the Gulf of Mexico Research Initiative. So just to get an idea of who I'm talking with, how many of you are already familiar with Louisiana Sea Grant or one of the Sea Grant programs? Okay, so a good number of you. Robert Twilley, our director, included in the audience here. So Sea Grant, I like to say that if there's a big body of water near you, whether it's a Great Lake or an ocean, or in our case, the Gulf of Mexico, there's probably a Sea Grant program near you. There are 33 programs nationwide, and the idea of Sea Grant is to enhance and conserve the practical use of our natural resources that are coming from either the ocean or the Great Lakes. So we're a non-advocacy organization, so that means we don't tell you what to think one way or another on an issue. We present you with the best available science and say, you're intelligent, you can make up your own mind what you think about it. So you're going to see a little bit of that tonight. We're university-based programs. The one in Louisiana is based at LSU in Baton Rouge. And we receive both federal funding from NOAA and also some money from the states as well. And we have three major areas of emphasis that you can see. We have three major areas of emphasis that you can see here, one of which is extension. And that's what we're doing tonight, extending the science out to people, from the lab out to people within the community who could use it either in decision making or for your livelihood. So there's 4 Sea Grant programs within the Gulf of Mexico, one in Texas, one in Louisiana that I've already mentioned. Mississippi and Alabama share a Sea Grant program, and then there's also one in Florida. So as I mentioned before, we are sponsored, the Oil Spill Outreach Program is sponsored by the Gulf of Mexico Research Initiative, or GOMRI. And what GOMRI is a $500 million 10-year investment by BP from non-penalty funds. So it's completely separate from the Restore Act. And it's managed by a 20-member independent research board, and also the Gulf of Mexico Alliance, or GOMA. And the goal of GOMRI is to really help people understand the consequences of Deepwater Horizon and come up with better ways to mitigate those consequences and better respond to them as well. So GOMRI has funded over $170 million worth of projects so far. And if you just look across the map in terms of research that's been funded, you can see these red little plus signs on the map are scientists who are leads on research projects surrounding Deepwater Horizon, and the blue dots are co-leads on projects. And there's also a few scientists working internationally as well. So many people all studying this disaster that happened. And GOMRI is really focused on 5 major areas of interest, and those sort of span really a broad range. These are some examples of topics that people have studied using GOMRI funds. And really what it breaks down into is 5 basic areas, and those are physical fate and transport of the oil and dispersants, the biological and chemical behavior and degradation of oil and dispersant mixtures, environmental effects, technological advances, and then finally public health. So these projects fall into those different categories. So a neat resource that you can go home and try tonight that I think all of you would probably be very interested in is GridC, the data cooperative that GOMRI has, and it's available on their website. Anyone who is funded with GOMRI money, part of the stipulation of receiving that funding is that they have to load their data onto this database. So it's available for anyone. You don't have to have a special username and password or anything like that. It's open to anyone, so it's very transparent. Another great resource that you may not be aware of is their publication database. And this is the same sort of setup as well. anyone who's received GOMRI funding and has had a project published, that publication, the abstract, and a link to it are listed on this webpage as well. So also available through the GOMRI website. So once you have research, you don't want it to just sit in the lab in someone's notebook collecting dust, right? You want to get it out into the community, into the hands of people who could use the information, right? So GOMRI has also supported a lot of outreach activities. And so those have included things like their research consortia who are out there doing projects also have their own outreach programs. They've funded things like the Consortium for Ocean Leadership. You might be familiar with them because they do the National Ocean Sciences Bowl. GOMRI has also funded things like the Smithsonian Ocean Portal, which is this really cool infographic. It's available online and it has these diagrams of the oil spill, and you can click on specific points within the diagram and it explains what was happening scientifically in a really cool and easy to understand kind of way. They've also funded programs like our Sea Grant Oil Spill Outreach Program, which I'm going to explain a little bit about. So, as I mentioned before, there are 4 Sea Grant programs across the Gulf of Mexico, and we've all banded together in this effort to help share oil spill science. So, I have 3 counterparts, one of whom is here tonight. So starting out in Texas, we've got Christine Hale, and she covers environmental health effects regarding the spill. I'm in Louisiana Sea Grant, and I tend to look more at the chemical and toxicological effects of the spill. Larissa Graham, who's here tonight, she does public health, and she wears 2 hats. She's housed in the state of Alabama and also covers the state of Mississippi. And then we have Monica Wilson in Florida, who looks at things like the physical transport of the oil and dispersants. And then we have a coordinator, Steve Sempier, who's at Mississippi, Alabama Sea Grant. So, as I mentioned before, there are other outreach efforts regarding the oil spill. Ours wasn't looking to compete with them, but really to complement the work that they're doing. So instead of maybe targeting a very general, broad audience or K through 12, what we're looking to do is to work with these target audiences listed here. These are audiences that Sea Grant has traditionally worked with, so we have like really strong ties in the community, and these are groups that we felt either, their livelihoods depend on the Gulf of Mexico, or they could really use this information in decision making. So we did a needs assessment, the 4 specialists, and we've spoken to date with maybe about 500 stakeholders from across the Gulf of Mexico and tried to understand what kinds of oil spill science questions they have. What were the things that they wish that they had known when the spill happened 5 years ago? What would have made their lives easier or better in terms of decision making or dealing with the spill itself? So from that, we've come up with a series of deliverables. So 12 bulletins, 12 science seminars and workshops. These are just some examples of some topics that we've come up with based on those needs assessments, those questions, and including the seminar this evening. So that brings us to the seminar, Understanding the Toxicity of Oil and Dispersant Mixtures and the Development of Alternatives. So I feel like based on the questions that we've gotten from people and just, those topics right there, to create a framework, you really have to ask the question, what happens if the shoreline does get oiled? Why do you use dispersant in the first place? So if we look at some of the work that came out of Deepwater Horizon, this is a great example. Everyone in Louisiana knows, and probably many people across the Gulf know as well, that Louisiana has an extremely high rate of land loss, right? So you don't want to do anything that's going to exacerbate that. On the left-hand panel, you can see this is an edge of a marsh, and it's nice and smooth and even, not eroded away in an unnatural looking fashion. And then if you look on this panel on the right here, you can see how it's really been carved away. It's really eroded. Just huge chunks are taken out of it. And that's an area that was heavily oiled during the spill, during the 2010 BP spill. So how does that happen in the first place? So if you look at the upper left-hand panel A, you can see that this is a marsh that's been heavily oiled. There's a lot of oil there. What happens is the oil actually starts to kill off the roots. Okay, and the roots are the roots of the plants in the marsh. And the roots of the plant in the marsh are what's really going to give that marsh that structural integrity. It's going to keep all the sediments, all the muds from running out into the water. So it's holding the marsh together. And if you don't have those roots, then you end up with a situation like this panel here on the right, panel B. And so you can sort of see there's this dark void. That's where there's no sediment. Up at the top, you can see that there's still some plants sort of hanging on. So I'm going to set this speaker down for a second. I'm sorry. So it's going to create this situation. Like imagine this is my forearm is the base of the marsh, and this top part of my hand is the top of the marsh. It's called an overhang. So you can see that the structure, maybe it's not very strong, right? And it's going to keep it getting hit by waves, and eventually it can fall because there's nothing supporting it underneath, right? So that's called an overhang. And what this group found was that overhangs in marshes that were heavily oiled, or there are actually significantly larger overhangs in marshes that were significantly oiled. And overhangs, as you can imagine, eventually lead to situations like C and D, where the marsh gets eroded away at a rapid rate. And that's what Gomery researchers found out, was that areas that are heavily oiled have higher rates of erosion. Remember, this is, Louisiana is an area that has already high rates of erosion. And they also found that the levels of PAHs, or polycyclic aromatic hydrocarbons, I'm not going to say that word again tonight, I'm just going to call them PAHs, but these chemicals are present in coal and oil and tar. And who in here likes barbecue? Show of hands, barbecue? Really, no more people than that? So if you've enjoyed barbecue recently, you've also enjoyed PAHs. They're those charred bits on the fat of your animal that you're eating. So those have PAHs too, right? So you're exposed to them in your everyday life, but there are certain ones that can be carcinogenic or cause health problems. And that's why, after the spill, there's been a lot of talk about those compounds. And so they actually found in this study that the PAH levels in sediments in those marshes where they were heavily oiled could be as high as 133 parts per million. And you might be saying, well, what's a part per million? A part per million, could everyone in the room just close their eyes for a moment? Play along. Okay, so an Olympic-sized swimming pool, in your mind, can you picture it? Okay, so you're picturing an Olympic-sized swimming pool. Now imagine 1.5 gallons, like a jug and a half of milk, except now it's oil, being poured into this Olympic-sized swimming pool. That's 1 ppm. So open your eyes and look at that number. So it's 133 ppm in the areas that were really heavily oiled. Some areas did have low oiling, and that's a much lower number, but so that gives you an idea. Okay, so what does that mean in historical context? You have these really high numbers of these compounds that I told you. Some of them are harmful, these PAHs. So a good example of an oil spill that happened in the past, Buzzards Bay, Massachusetts in 1969. There was a ship that ended up spilling a lot of oil. And the consequence, a lot of marsh was oiled, dead animals like fish, horseshoe crabs, regular crabs. So that was back in the 60s. These photos were taken at that time. Then, as you can see in this photo on the right, in 2005, it looks like conditions are back to normal. It's a beautiful, healthy, vibrant-looking marsh, right? But what about chemically? What about the animals there? So there's this interesting study that was done back in 2007, and the researchers decided to look at fiddler crabs and how oil that was left behind in the sediments affected their behavior and their escape patterns. And what they found actually was that the way that these animals, these little fiddler crabs that are an important prey item, important to the ecology of a marsh system, that they actually weren't able to burrow as deeply because they were trying to avoid areas that had high levels of oil, high levels of these PAHs, these toxic compounds. So if you look at this graph, you can see that the burrow depth for an unoiled site versus an oiled site is quite different. It's much deeper in unoiled sites. You can also see that the shape of these burrows. So if we look at the left-hand panel photograph, the oiled sites, you can see that the shape of the burrows is a little bit curved, and the researchers actually found that this is where they're trying to avoid oil. They're a little curved. They're also pretty small. They're about half the size to a third of the size of these much longer burrows taken from unoiled areas. And just in case you're wondering how they got these burrows so exact, what they did was they went out into the marsh, they found the fiddler crab burrows, and they poured plaster of Paris into the holes, let them harden, and then they could pull them out and really quantitatively measure what was happening. So I thought that was pretty cool. Also, they found that the PAH levels were around 134 parts per million. So again, remember that Olympic-sized swimming pool, and a jug and a half is 1 part per million. So really high levels still in 2007, and this spill happened in 1969. They also found that the PAHs had a neurotoxic effect on the crabs as well. It affected their escape response. So, if you're a small little crab and you need to get away from a predator, how quickly you can run away is really important, right? So if you look at this graph, they took crabs from an oiled area, and they either fed them bits of marsh that were from an unoiled source or an oiled source. And on this height of the bar indicates how long it took them to run away from a perceived predator. They created a disc that would swing that would startle these fiddler crabs. So they monitored how quickly it would take them to realize, hey, I should run away. And as you can see from the graph, the ones that were fed the oiled sediments took a significantly longer time to react to this potential predator that could eat them. And remember, this happened in the 60s, and you're still seeing the effect today. So another example of how sediments from oiled areas can affect animals. If we take Deepwater Horizon as an example. So killifish, maybe you're familiar with that as a bait fish. Do we have any fishermen in the room tonight? Anyone that likes to go fishing? Yeah, okay, so you know that this is an important bait fish. So economically important. They're also important ecologically. In terms of animals, you can think of them as more of a base of the food chain kind of an animal. So this is a photograph of them as an adult. And this study by Gomery researchers actually looked at their embryos. The little baby Gulf killifish, while they're still in their little eggs, you can see them developing. They've got an eye, they've got, I don't know, maybe a tail. You can see the heartbeat. So what they did was they took sediments after Deepwater Horizon, and they took them from areas that were unoiled in 2011. So 2 sites in 2011, they took sediment, and then they took sediment from Grand Terre, a site in Louisiana that was badly oiled. And they took sediments from that oiled area in 2010 and 2011. And so they looked at what would happen if you took these sediments, you took them back to the lab, you put them into individual containers, and then you put these little fish embryos into each different container, or into separate containers, and you looked at which ones survived, which ones didn't. What type of sediment were they exposed to? Was it from an unoiled area or an oiled area? They looked at hatching, heart rate. So they found that the mortality, or how many of those that died, the percentage, as you can see from looking at this graph, it's pretty similar. There weren't any significant differences there, oiled versus unoiled. But then when they looked at how many actually hatched, that's a big difference. So the ones that were exposed to sediments from Grand Terre from right after the spill in 2010, a significantly lower portion of them hatched than those that were even exposed to sediments from a year after the spill from either Grand Terre or an unoiled source. Then if you look at heart rate, you see something even more interesting, that it not only affected their heart rate, they have a reduced heart rate if exposed to oiled sediments from 2010 right after the spill, but even sediments from that same site a year later were still impacting the heart rate of those fish. So you can imagine the heart is an important body organ, not just for humans, but for fish as well. So that's pretty incredible. So now you have some ideas of, maybe why emergency responders might choose to use dispersants as a response to an oil spill. But what are dispersants, right? Just so we're all on the same page, I'm sure that many of you are already very familiar with them. But let's just go through it again. What are dispersants? So historically, we didn't always use Corexit. The first incidence of dispersant being used was the Torrey Canyon spill off the coast of Cornwall, England, back in the late 60s. And you can see from this map that around here is where the ship went aground, and oil coated this whole southwestern tip of England. The oil traveled down, actually covered some of the shoreline of France as well. And the dispersants that they used were created from kerosene as a solvent and a ship degreaser, something that was used to clean ships' engine rooms. So not necessarily something that's very environmentally friendly, as you can imagine. And the result was that it actually killed off almost all the life in these areas. So you ended up with these areas that were totally devoid of things like razor clams and barnacles and limpets, all these animals that you would typically expect to find on a shoreline. So there was a study that was done in 1968, and they actually looked at how this might affect, because you might think, well, okay, everything dies off. Eventually, it will come back to the same area, right? So 1968, a year after the spill, some researchers took a look to see what the community structure was like, and they found that it was completely different from the way it was before the spill. And so if you look at this upper panel, you can see there's a lot of green algae and brown and red algae. These are sort of the opportunistic species of algae, and lots of laminaria. Everything was really overgrown by all the plants on the rocks. And the reason why these rocks were so overgrown with plant species was because there were no animals to graze the algae. So there was a total shift in the community structure. Then if you look at this bottom panel, in 1977, researchers came back again almost 10 years after the spill, and they found that the herbivores, the animals that eat plants, had returned to the area. There were more, if you can tell from the diagram, more barnacles and limpets and more types of algae, and the laminaria was more under control. It wasn't such an overgrowth of everything, but they found that actually the community structure still wasn't quite the same as it was pre-oil spill, and this is about 10 years afterward. So this is a really nasty case of dispersant use, and it was the first case of dispersant use. So obviously things have shifted since then. One of our dispersants that we use is Corexit, and as you can imagine, we use it to prevent the oiling of shorelines and animals. It is composed of a solvent and a surfactant, and some of the components in Corexit are things that you might find even in your food or your personal care products. There's a portion that's oil-loving and a portion, water-loving, and a portion that's oil-loving. You can see it kind of almost looks like a lollipop or a push pin. And just so you know, the water-loving portion really doesn't want to be in oil, and the oil-loving portion really doesn't want to be in water. And the way that it works is that, say that you have an oil slick, it's going to help break up the oil into little droplets, right? Which wave action can do that too, right? But with wave action, eventually those oil droplets are going to get back together and recreate a slick. So you want to create a situation where that slick doesn't have a chance to reform. And so that's where the surfactants come in, and they sort of stick in those oil droplets, almost like push pins in a pin cushion, right? And so once they envelope those individual oil droplets, it's intended that they won't reform the slick, and that's going to enhance bacterial degradation. The bacteria are going to be able to eat these little droplets of oil easier than they would a gigantic slick. So that sort of gives you a background for what our two speakers, Drs. Chris Green and Andrew Nyman, are going to talk about. They're going to go into the effects of oil and dispersant mixtures, a really specific study that they've done together that sort of looks at how environmental conditions can affect the toxicity of oil dispersant mixtures. And also, Dr. Nyman will talk about some alternatives and really put everything in an even broader framework. So, yeah, I'd like to welcome Dr. Christopher Green.
There's a mic and a...
Mm-hmm.
All right. I can hold it. I can hold it, yeah. I get that. Maybe I can't get that. There we go. Okay. Sure. Thank you. Now I'm going to grab the, so I have a microphone for our online people, and I've got my pointer. So I think we're ready to go. Thank you guys. Thank you everyone for giving me the opportunity to speak here in this forum, first off. And as you know, Emily did a really good job introducing, so we're going to talk about a couple of different published studies in the literature that represent a basic introduction to toxicology, specifically with the oil spill. So we're going to see, you know, a pretty good introduction into toxicology in the context of a few specific things. And some of the things I want to talk about is time, and I want to talk about salinity. As we know, our estuarine environment down on the coast, it can change. It can be influenced by our fresh water, by our salt water, and seasonally and even through tidal cycles, it can change. It's a very dynamic location. It changes a lot. So, you know, when we look at toxicology, the basic principles of toxicology are the dose, the amount of a chemical, or in terms of oil, a mixture of chemicals, and the dose and time, so the amount of that exposure. So with respect to oil and biological components, a lot of these things are going to interact across time, and that exposure is going to alter how those animals may react. And also, with salinity, salinity changes how those animals regulate things in and outside of their body. It can regulate spawning cycles. It can also have chemical influences, so it can really kind of alter the way that these chemicals like dispersants and oils are available to get inside the body. When we look at those things of dose, of time, of these things in our environment like salinity, we have different availability. And so I'm going to focus on oil, surfactant, and then a mixture of oil and surfactants, because oil and surfactants are going to dramatically change the amount of the compounds that are in oil in the water. And I have some graphs to demonstrate the whole multitude of chemicals that make up crude oil. And then finally, responses. Now, for just basic principles here, we're going to talk about things like mortality. But keep in mind there are other responses. Responses like what Emily talked about with those developing babies, or those adults, those sexually mature animals that are investing in things like sperm and eggs. And then the consequences past that, whether it be the peat, these other larvae, or the way we utilize our resource. So first off, I want to introduce really the star of our lab, the Gulf killifish. We utilize the Gulf killifish for a number of different things. I started utilizing this animal here as it is economically important for angling in this state and across the Gulf of Mexico. So it's a long spawning season animal. It's going to spawn all throughout the late spring, summer, and even have some fall spawning as well, according to our research. It's prey for a lot of good sport fish. So if you go fishing on the coast, shrimp, Gulf killifish, things like that are a really desired prey item for anglers. And also, it's abundant. I think Emily indicated this a little bit. It eats a lot of things in our estuaries, but it also gets eaten by a lot of things. So it plays a really important role in terms of moving energy around in this environment. Now, in terms of a little bit more sciencey aspect, we maintain a number of different captive populations of this fish. This fish is distributed all throughout the Gulf of Mexico and down even into Mexico. We actually have multiple populations all the way from Florida up through where we're at right now and even into Texas that we maintain in our lab. So we have a captive group that spawns readily. Reliable reproduction. We can reproduce these guys, and as we saw with the babies that Emily talked about, we can do different experiments with these guys because we know what kind of good conditions to make them spawn in, and we can use them as a good experimental organism for a number of different possible responses. One of the other things, if you look out here into the marsh, and the tidal influence is coming in and out of here, a lot of fishes that are out in the open waters, whether they're larval fishes or whether they're full adults, they have the ability to move around in a large expanse. And I don't know what that means. But the point is, it's okay. The point is, these guys hang out in a very, very small area. So they're going to loiter around in the marsh grasses and things, specifically in areas that are really specific to particular amounts of oiling. They're not going to migrate very, very far distances, say like a whale or a tuna or something like that. So what they're experiencing in the marsh is very site-specific. Can I see my next slide to give me just real quick? You don't have to bring it up, I just see what. Okay, so I just wanted to, sorry about the technical difficulties, folks, but we'll work past that. I wanted to talk about a published study that looked at creating a small environment that I call a mesocosm in a greenhouse. So what we did is we had small mesocosm environments, so probably around 20 to 25 gallons, where we took marsh sediment, about 5 gallons of marsh sediment, and we set these little bitty environments up like little bitty swimming pools. We then took different amounts of Corexit, 9500, oil and Corexit in specific mixtures, and then what I'd call a control, or with none of those things, just regular salty water. And we were able to, if you saw this graphic earlier, we were doing some pre-weathering. So we would mix the oil, the Corexit, and the oil and Corexit, which is missing right here on the sidebar. We would mix that to create a mixture that we'd then be able to add in to these mesocosms. I'm going to pause just here for a little bit while we get our connections up. I don't think anybody's got any other appointments after this, do they? This is the best I could do. I could talk for a little bit, and then I had to pause because I've got some other graphics that might be a little bit better in this show. Connection is timed out. So we have a couple of minutes, and based on a couple of the things. I think we've got a few minutes while they get reconnected. Does anybody have any questions about the Gulf killifish or basic, oil questions? Maybe something simple we could field in the couple minutes we might have here.
What is a mesocosm? You said a micro. I could imagine a microcosm.
Yeah. So it's the jargony word for a small replicated environment. Yes. So what we do is we have in a greenhouse, we want to replicate small environments that we can weather this oil in, where we have a layer of sediment, and we'll have in that sediment we'll have a hypoxic and an anoxic layer of sediment in there, and it'll be we'll regulate the temperature, we'll regulate a lot of different factors environmentally, so that we can place oil in that tank, in that system, and let it hang out to as if it was just in the water or around some of the marsh grasses and things like that. So it's a lab way. One of the things, a lot of you guys might have questions about specific things in the environment, and one of the things that we have in here is I'm removing a lot of the variables in that environment. So if we think of the mesocosm as an aquarium with water, with oil, say a small oil slick on top of it, some of the things we don't have are, some of the big wave action that might disrupt that and break it down a little bit further as the tide goes in and out on the marsh. We might have, wide swings in different plumes of algae communities and things like that. So we're removing a lot of those environmental variables, which gives us a lot more control.
They want to hear what you're saying.
Oh, okay. Well, I put it down. So I'm sorry. I was just ad-libbing here. So, as it were. We're working on it. It's being worked on right now. So anyway, we can keep talking about some of these responses if that's okay. We can look through these pictures. But the point is, we wanted to set up these small aquariums with sediment at different environmental salinities that we might see in the marsh, to look at how that over time might change different parameters that might be of importance to, say, the toxicity of those combinations. So we set up these mesocosms at 4, 12, and 18 parts per thousand. And just to give you a reference, parts per thousand is grams per liter. So the salinity of the ocean is roughly 35 grams per liter, 35 parts per thousand, 35, 34. So we were working in conditions that were more, lower salinity that we might have as we're, heading towards the coast. So say if we wanted to replicate, say, full salt, we'd be at 34 parts per thousand. We were at 4, 12, and 18, some of the salinities that we've encountered in some of our sample sites along the coast, specifically for some of the areas that had been affected by the oil spill. So what we did is, once we set up those different salinities, we then took and we would put our treated mixtures, Corexit 9500 or Corexit and oil, or just oil, and we'd put it in these mesocosms and we'd wait. We would sit and we'd then collect the water from underneath the slick or in that mesocosm at 1 week, 4 weeks, and 16 weeks after the application of that water. We would then do some acute bioassays, so acute toxicity measures. We would take that water, so say we had a bucket here, we would take that water out and we would dilute it in very specific concentrations by half, and then by half again, and then by half again, and half again. So we had 5 different concentrations of a diluted proportion of that mesocosm. So if the mesocosm water represented no dilution, or 1, the next one in line would be what? 0.5, and then 0.25. So we would be diluting it such that the chemicals that were represented in that mesocosm would be then across a known or predicted value. We could then assess the amount of PAHs in that water or Corexit compounds, and we would see a gradient across there from high concentration to low concentration. Now, we would take small juvenile or teenage Gulf killifish that were roughly about this big. So nothing you really want to put on a hook and really catch, because they're kind of tiny, but they're right before sexual maturity, and they represented a really good uniform population. I started working with Gulf killifish specifically because of my interest in it as a bait fish, and so getting a real uniform group was a pretty big importance. So we had these teenage Gulf killifish, and we put them in replicated jars with this water with a little bit of air bubbling in it. And we leave them in that water for a specific amount of time. So we had a dose, that 1.5, you know, that gradient, and we'd have a time. We'd have 96 hours or 4 days. So if we put those teenage killifish in that thing of water on Monday, they would come out of that water on Friday. And every 24 hours, we'd go look at our things, and we'd change out the old water and put new, treatment water in, and we'd measure the mortality. So over time, throughout those 4 days, we'd measure that mortality. And we created graphs to detail the mortality that you would have from these different treatments of oiled water and weathered oil. So are we ready to get back? Okay, cool.
Thank you all very much for being so patient.
No problem. Pardon? Oh, okay, cool. I need to jump over here, and I need to start I have an animation for us that I think is going to be, hopefully it'll, where's it? Wrong one. Can you click on that? So this is a video of straight oil, just to demonstrate some of the difference in the physical properties. This is some of our pre-weathered oil being put in a mesocosm. This represents roughly 40% coverage of the area. And then this is representing our Corexit oil mixture. And Emily talked about the formation of those small particles in there. You can really see a dramatic difference in terms of the availability of that oil getting into the water. And so that's going to be, as it's intended to be breaking down and things like that. So we can see that. It's really, it's a very noticeable response. So some of this stuff, while we were waiting, we've already covered this. So some of you guys have already heard this. Actually, everybody did. This is the graphical representation of what I've been talking about. So down here I have the salinity, 4, 12, and 18 ppt, which is, I didn't do the math to figure out how much was the percentage of the ocean. I apologize. But 34 is roughly 100%. So if you look and divide that, this is 12 ppt is roughly 1/3 of the salinity of the ocean. We have time. Each is weathered for 1, 4, and 16 weeks. And then again, as a reminder, here are our treatments: control, oil, Corexit, and Corexit and oil. Now, we talked about polycyclic aromatic hydrocarbons, and crude oil is a huge mixture of compounds. It is not just one thing. This is just a small number of those. This is 72 of the main constituents of oils. But, this is going to blow your mind, there are literally thousands of these compounds, and they continually change and get modified and things. We're continually finding new ways to measure polycyclic aromatic hydrocarbons, and this is just the main categories. The take-home message with this is that when we looked at the amount of those PAHs that were in those mesocosms, the top being the ones that had a slick maintained on top, just oil, and then the bottom one being that chemically dispersed oil, is really apparent that no matter across 1, 4, and 16 weeks, the availability, the amount of those things that are in what scientists call the water accommodated fraction, or the water portion of that mesocosm, is really, really high. You can see families of these here. And these families are based on different weights, different carbon bond and chain things. But again, this is only 72 compounds, and there's a whole host of these other ones too. So keep that in mind. We talked a little bit about toxicity. We talked about dose and time. And this is the graph that we look at when we create an acute toxicity bioassay. So on the bottom here, we have the dose. So that would be our proportion of dilutions. So this is the highest dose, followed by a low dose. This isn't actual numbers, it's just for example. And then effect. So there's some kind of an effect from this dose over here. And for the purposes of this, we're talking about acute toxicity. And so we're talking about the number of dead teenage Gulf killifish throughout that 4-day period. And what it creates generally is this curve right here. And toxicologists, they want to use that part of the response that's the tightest, the least amount of variability. And that is found statistically right here at 50%. So the LD50 is the dose that produces an effect in 50% of the population within that specified amount of time. And that's the most least amount of variability, is in that 50% mark right there. Now, there's something underneath here. I'm not addressing this, but underneath toxicity, before you even see individuals dying, you see what's known as the lowest observable adverse effect level. And that's the lowest concentration that produces a detectable response. Now, today, when we're talking about toxicity, we're talking about things that, fish that die over, say, a 4-day period. But fish that do survive, they've got things in their livers, in their brains, and the rest of their bodies that are constantly dealing with this oil or any other kind of compounds that they see in their daily lives. And, the effects of that is just as important as mortality. So here's our 4-day exposures. These are those little containers that we used. We changed the water out every day. Here's our Gulf killifish teenagers up here at the top right. Here's our dilutions I talked about. So we had just straight from the mesocosm, and then we cut it by different proportions, half by each time. And we recorded daily mortality. Collected survivors, and we collected tissues. We collected brain tissue and fillet tissue and liver tissue, because just like, if we go out and we eat some food, or we get exposed to, poison ivy or something, their bodies are having to deal with the chemicals in their system. And so we wanted to monitor that. We created these mortality curves. So this represents our highest salinity, 18 parts per thousand. Along this axis is the dilution. So as we ride from the left to the right, we have a high dilution to a low dilution. So we actually have the highest concentration, that's straight from the mesocosm, and then as you go this way, there's less pHs, less chemicals. Across here is the number of survival in that group. When we look at the purple or blue, and the green line corrects it, and oil corrects it, we see that it produced a strong amount of mortality in these dilutions here. After 28 days or 4 weeks, we didn't see a lot of acute toxicity. So we saw this dashed line represents what we had in terms of survival from animals just hanging out in regular, clean 18 ppt water. Very, very similar values, very early on, between 7 days and 28 days. Like the last one, at 112 days, we had reduced acute toxicity, really similar to control, so we didn't really see an acute toxic response. At 4 ppt, in contrast, our lowest salinity, we saw that oil and Corexit produced a really strong acute toxicity curve right in here. So just mesocosm water, if we just took that water out, that water killed 100% of those animals. When we looked at the value of Corexit, this was actually, we had looked at this Corexit value here is pretty much in line with what we see in the literature. So this is real predictable. We knew that what we added in terms of Corexit would produce this response. When we look at 28 days, however, Corexit is higher up, indicating it is slightly less toxic. It's reduced slightly. However, oil and Corexit is still producing 100% mortality at that just right out of the mesocosm concentration. And then when we look after 112 days, we see not a whole lot of acute toxicity here. So we have a lot of animals surviving. That doesn't mean that there aren't other things going on inside their bodies, and we have some tissues collected that we're analyzing right now for that work.
Can I ask a question?
Oh, yeah. Yes.
When you take the on the surface or on the bottom, I mean.
Yeah.
That's probably why it doesn't cause the infection.
I will.
because The fish is not exposed to it. Right?
Correct. So what we have is this red line up here. The question for the people at home, this red line up here, the oil, just oil, it's not producing a mortality effect. It's just up at the top or it's down in the sediment. So it's not producing acute toxicity, and that is absolutely correct. What's happening is we had very little acute toxicity from oil alone. That oil is sitting on the slick. Now, if this was out, and this ties into your mesocosm question, if this was out in the wild, what would be happening on a lunar period in the marsh? And I'm waving my hand up and down. Huh? Exactly, tidal exchange. And tides, the winds, are active things that break up these oil slicks. And so that's one thing we don't have in this lab study is the active, tidal influence and the breaking down of that oil isn't occurring in just this kind of mesocosm static environment. So what you're seeing is very little transference into the oil. And you saw that graph earlier with the dispersed and non-dispersed oil. You're getting a completely different profile of PAHs that you would. So hence, you're not seeing that acute toxicity. And so that non-dispersed oil is not breaking up. Now, if we did this study out in the environment, if we wanted to dirty up a bunch of stuff, which I don't want to do, we would see tidal influence and tidal and other wind influences breaking up this oil and possibly making survival possibly decrease a little bit. That's a guess, but probably a pretty good guess. Now, at 16 weeks at the intermediate salinity, we see some variable mortality in here, but again, we don't get the same kind of trends we saw here at 12 ppt and at those other salinities where we saw acute toxicity even at 28 days in some of those other salinities. So what did we really see from this? The non-dispersed oil really lacked acute toxicity, and it's predominantly because of its availability and getting those toxic pHs in the water, okay? The salinity of the water definitely influenced the toxicity of Corexit 9500. Dr. Andy Nyman is going to address some of our collaborative research to address some of those issues. A lot of that has to do with the way those fish and other aquatic organisms are balancing salts and other ions inside and outside their body. So it has a lot to do with how surfactants act at the gills and inside their intestines and stuff like that. At the low salinity environments, we saw greater persistence of acute toxicity. So at the lower salinity environment, we saw that the acute toxicity we observed at 1 week hanged on the longest into that 28-day period, although it didn't maintain itself at that 112-day period. So I want to point out, this is about the end of when I'm talking, and we're going to let Andy talk specifically about surfactants and biological surfactants. I want to point out something. I've talked and introduced stuff about acute toxicity. And when we're talking about that, we're talking specifically about, mortality. We're not addressing, in this talk, some other things that we're currently studying in the lab, like sublethal toxicity. What's going on inside these baby killifish during the development of their brain or their hearts or their liver tissue? What are the implications for oil exposure and metrics like, say, the length of this guy, this red line here, or the amount of yolk, the egg yolk that you might have on your egg in your breakfast? That's really, really important. A lot of that stuff is guided by those adults. And some of the stuff that we have going on outside of the acute toxicity work is we're starting to look more at the adult toxicity and what we would call multiple generations. So I talked about the teenage killifish. What if the survivors of those teenage killifish reached sexual maturity, adulthood, and then produced their own babies themselves? So that's some of the things we're looking at right now is the multi-generational effects of that oil exposure. And we're also looking in specific things inside that animal. We're taking out livers, we're taking out brain tissue, gill tissue. A lot of those parts of the body that are interacting directly with the environment. So this is the same graphic I had before. It didn't really kind of introduce and get us thinking about time and salinity, but also in how that really relates to availability in our environment. And as we see, as we continue to see during different storm events, the resurgence of oil along beaches. I was at Grand Terre 3 weeks ago. And then those responses, whether it be young, sexually mature adults, our species of concern and things like that. Some of the stuff that Andy's going to talk to you about, microbes, or, and I think this is really important, like some of you guys in this room, how we use and preserve and get the most out of our environment, and are also kind of the best stewards of our environment. So I know I probably didn't answer everybody's questions, and I'm sorry for that, but we're going to have a period for questions after Andy's talk. We'll come back up here and talk a little bit more. Some of this funding, actually the mesocosm work came from Gomery, specifically some of the other work that we've branched off of right now for the adults, for the multiple generation work, is coming from the National Science Foundation and Oceans and Human Health, part of the National Institutes of Health. I'd like to thank you guys for your time, and I'll be back up here after Andy's talk to talk with us, have a conversation.
All right. And y'all, how y'all doing on the telephone?
They know not to answer.
Much easier to hear.
Say that again.
Much easier to hear now.
All right, I'm gonna try to keep it right up here.
Okay, thank you.
You're welcome.
You still waiting for the next speaker?
Yeah, they're loading up my presentation right now. I'm gonna try to figure out this controller for a second. Let's see. Which one's it?
Can I ask a question while you're doing that?
And this is your laser.
Yeah, go ahead and ask a question.
Okay. Talked about killifish and having a small area that they operated in. Could you speak to all the other organisms that we are concerned about, and whether they are more typical of the killifish or more typical of animals that can go far and wide and away from the spills, if it was possible?
All right, I'm going to relay your question to our audience here, and then I'll take a stab at answering. What's your question? The question was, all right, so they understood that the killifish hangs out at one spot for a long time, so it's kind of representative of that location. But what about other animals in the Gulf? Do they hang out at the same location all the time, and can you kind of relate their health to that location? My understanding is that many of the species that we're interested in range far and wide. The tuna, the whales, a lot of our birds, they travel and visit many places. And so it would be a lot more difficult to detect an effect of one of those animals visiting multiple locations across a year or two. So I think those animals it's a lot harder to figure out, to detect an effect. Chris, you got anything to add to that?
Well, yeah, one of the things with the open water, you have a lot of species, a lot of high-profile species are going to move around, things like that. And you're right. What happens is you want to look where the oil goes. It'll be the sediment, other components.
Chris pointed out, for those of you on the line, that in addition to the animals moving around, the oil is also moving around out in the Gulf. And so it gets buried in the sediment for a while at a place, but then can be mobilized after a storm. And the oil's moving, the animal's moving. So detecting the effects of oil and dispersants on animals in the Gulf of Mexico is a very challenging job, and one that neither Chris or I do. We always work in the greenhouse or in the laboratory where we have these aquarium where we can keep the salinity constant for 2 or 3 months while we let the oil biodegrade. And that way we can infer the effects of salinity on these processes. Your question is very important, but one that we really can't answer with the tuna and those bigger animals. All right, I'm going to go ahead and, did that answer your question?
Yes, I think so.
Thank you. All right, I'm going to go ahead and move into,
Was that, are we talking about like all species? Are we talking about things with economic value, or who's
Could we
looking at?
please hold all questions
What?
till the very end?
we're going to come back to your question. Hold that till after my presentation, and then we'll revisit that. All right, I'm going to go ahead and, Chris gave you an example of one experiment, and I hope you got a sense of the amount of effort that goes into being able to say, like what he said at the end, the dispersed oil was more toxic than the undispersed oil, and that toxicity persisted longer. That's so easy to sum up and say, but I hope you get a sense of all the aquarium that had to be set up with the same amounts of sediment. The water's the same except for the salinity. We've got the oil, you got all those fish you have to raise and expose them at the right, raise them at the right age. It's an awful lot of work to say a sentence like that. What I'm going to do is, I've lost my microphone? Okay. All right, I'll keep it right by my mouth. Thank you. What I'm going to do now is I'm going to say a lot based on a lot of research, and I'm not going to give you any details on how that research was done. I'm going to cover a lot of ground. That's my intent here. So the background information, what I want to remind you is how many things are in crude oil. Chris said this, and I'm going to say it again because it's extremely important. There are tens of thousands of different compounds in crude oil. I've heard in presentations up to 750,000. I found citations up to 36,000. I'm not sure why I've heard that difference, but there's a whole lot more compounds in oil than we can chemically identify, and certainly even more, and even fewer of them we can identify and measure their concentration. And that turns out to be a very challenging part of the problem we face. We can measure about 150 of those compounds. As time goes on, the chemists are getting better at identifying a compound and determining its concentration. We're up to about 150, and you hear this during the oil spill. Ed Overton was on TV a lot, and you hear about oil fingerprinting. This is the fingerprinting. It's GC, mass spec, type work, and I'll refer it to as fingerprinting. It's kind of it's the state of the art in oil spill response, but it's a big difference from being able to fingerprint and say we got these 150 compounds and recognizing that there's at least 36,000, maybe 750,000 compounds out there in the crude oil. The dispersant is a mixture of chemicals. So again, it's not a single compound. It's a whole mixture of compounds designed to break up oil and dissolve it in water. All right, so I'm going to review first kind of an overview of the effects of oil and dispersed oil on our coastal ecosystems, and then I'm going to start talking, I'll move into alternative dispersants. But first of all, I'm going to talk about marsh grass, and then I'll talk about the microbes that live in the mud, and then I'll come back to the animals where Chris was, and then we'll move into the next generation, or search for a new dispersant. Well, why I care about the marsh grasses? The grasses are important because they're the base of the food web. They make, they do the photosynthesis. They make the litter that the worms eat, that the shrimp eats the worm, the trout eat the shrimp. That's the base of the food web. If you kill the grass, you're going to have less energy going in the food web. The above ground parts are structurally important because this is where, again, some animals eat this directly, but also it's important, particularly during winter, to provide cover from cold weather for a lot of wildlife. The below ground parts are especially important. Emily mentioned up front about the roots. The live roots are what give these soils their strength, and that's what makes the soils resistant to erosion. If you kill the roots, you reduce the strength of soil, and you therefore increase the erosion rate. The below ground part, the roots are also important because it builds peat. You think about trees, they grow, they get bigger and bigger around all the time. They lay down wood, layers of wood. You can cut a tree down and see those rings. A marsh is doing something similar, but you're getting sheets of root building up on the marsh year after year. And this is how a lot of our marshes offset subsidence and sea level rise. So if you stress the plants and just reduce the production of roots, you're potentially increasing the erosion rate and you're slowing the rate at which they build up. You're slowing their ability to offset subsidence and sea level rise. And then the marshes drown and convert to open water. All right, here's a picture of some roseau cane down at the mouth of the river just during the spill. And you see a bunch of dead leaves. The oil has come in on the bottom of this plant. It appears the oil has been moved up the plant and killed all the leaves in the plant. And all the leaves are dead. And yet, you see these new green leaves coming out. I'd seen this in the greenhouse 10 years before with a different species. It really shocked me that there are a lot of marsh plants, a lot of marsh grass, a lot of marsh plants are very tolerant to crude oil. All the leaves die and it just makes new ones. Now they can't do this over and over again. It's going to run out of energy reserves after a while. But many marsh grasses are tolerant to crude oil. And there's been a lot of work looking at dozens of research projects looking at the effects of crude oil on marsh grasses. And we can go ahead and summarize that. There's too many. I'm not going to go into any specific studies now. But the pattern's clear. The oil on the plant is a whole lot less of a problem than the oil on the roots. The roots are where the plants are uptaking stuff to bring to the rest of the plant. They're also important. They have a hard job getting oxygen down to them. The upper part of the plant basically is a snorkel for the roots. That's what makes a wetland plant. And the oil getting on the roots is a much worse of a problem for the plant than oil getting on the stems and leaves. The other pattern that we also see from looking at the response of plants to different concentrations of oil and then looking at how it happens out in the field is that I now think of it as 3 zones of concentration in the field. This red zone here where there's enough oil in the soil to kill the vegetation and prevent new vegetation from moving in for months and maybe even years at a time. Beyond that zone, you can often find a larger zone where the concentration of oil is lower. The adult plants can survive, but they can't reproduce. And beyond that's an even larger zone, the light green here. Battery's dying. The light green, where the plants have a little stress, but then after a while they do recover and they come back. And that's the way I think about this now. And this and it's really all about the oil in the soil. Well, not all about. It's a lot more important where you get oil in the soil than just on the top of the plant. There's been enough work by enough researchers now that we can actually rank the sensitivity of different plant species to oil. Not all plants have the same tolerance to crude oil. Some are more tolerant than others. All right. The map that I'm showing you here is a map of the different marsh types in coastal Louisiana. Marsh type is a group of plants that we recognize that occur in different salinity zones. Like the purple color up there is the bald cypress swamps. You come further out, the dark green color, that's what we call a fresh marsh. Come on down, there's a light green color. Most of us refer to that as intermediate marsh. Then you get to the red, which is the brackish, and the yellow one is the salt marsh. As you move from low salinity to high salinity, the number of species in the plant community drops tremendously. And by the time you get to salt marsh, there's really only one species that dominates them. When you get up in the fresh marsh, it's very patchy. You've got many species, and you can have, there are several plants that may dominate. One species might dominate here for a couple hundred acres. You move over a mile or two, it might be dominated by a different species. And there's a lot more biodiversity in the fresh marshes. But if we look at the rankings of these, you see that our most sensitive plant species is Phragmites, the one that I showed you. It also occurs at the very end of the Mississippi River. You might think that you look at the map of Louisiana, and that sticks the farthest out in the Gulf of Mexico. You'd think that'd be the saltiest plant communities we have, but that's not true. Those are dominated by fresher communities because there's not enough salt to stress these plants, and the salt plants can't make it up in there. So the Phragmites, some of the first areas that get hit by oil, are also some of our most sensitive to oil. The Spartina patens, which dominates our brackish and our intermediate marshes. Many of you will know it as wiregrass. I forgot to mention the Phragmites is all roseau-cane. If you build a duck blind, you're building it out of roseau-cane. That's our most sensitive. The wiregrass, the Spartina patens, seems to be our next most sensitive. And then you get to kind of a tie between Panicum hematomon, Pyphene, it's a fresh marsh plant, maiden cane, are also, and it's kind of tied up with the salt marsh plant, Spartina alterniflora, the oyster grass. Many people call it oyster grass because when you start seeing it, you know you're salty enough to find oysters in the water. And our most tolerant species is the bull tongue, a fresh marsh plant that just seems to tolerate crude oil very well for some reason. Notice the fresh marsh has our most sensitive and our most resistant species in it. There's no relationship between the sensitivity of or the tolerance of these species to flooding stress and oil, or between their tolerance to salinity stress and oil. These effects are unrelated, it turns out. And this is useful information. If you're a spill response person, you can use your ability to identify plants to know where to put boom. If you don't have enough boom to cover everything, what do you want to do? You want to put the boom in front of the most sensitive species you have on your piece of real estate. Also might help explain why you might see more effects farther inland than out on the coast. Notice the salt marsh plant is more tolerant than our Spartina patens, our brackish marsh plant. So it's possible to have less oil do more damage further inland because the plant species differ in their sensitivity to oil. All right, I want to move on and say a few things about marsh mud. Marsh mud's really cool. It really, really is. I never would have thought this about 30 years ago, but marsh mud is some really cool stuff. Why? And everyone should care about marsh mud. Why I care about marsh mud? Well, because it rots. Rotting is really cool stuff, it turns out. It's really important in the environment. If any of you garden and you compost, you're using that rotting of that material to release nutrients, nitrogen and phosphorus primarily, for your tomato plants, whatever it is you're growing. Out in the marsh, that same process is what supplies a lot of nutrients to marsh vegetation. So you can imagine if you were to kill all the bacteria that decomposes peat, you're going to reduce nutrient availability to the plants that make the roots that hold everything together. So we don't want to have an oil spill or a dispersant or anything really come in and reduce the rate at which the peat is decomposed by the bacteria. But also, these bacteria are eating this peat. They're eating the thing that builds the elevation that offsets subsidence and sea level rise. So you don't want to speed this up either. You might say, well, let's speed it up and get more nitrogen and phosphorus available to plants. But if you do that, you can actually eat up the thing that supports the surface elevation of the marsh. So you don't want to slow this down either. We want to keep it the way it is. I wouldn't want to change this because I wouldn't know which way it would go. You know, a lot of these marshes are barely hanging on as it is. They're barely keeping up with subsidence and sea level rise. I certainly don't want to accelerate decomposition on these things. These microorganisms that live in the soil also eat the oil. Now, they don't take bites out of it. I'm going to say eat. I do want to make clear what they're doing is they're secreting enzymes that break down the oil into molecules small enough for them to absorb and then eat and get their energy that way. But this is a community that eats the oil. So you don't want to put something in here that's going to kill the bacteria that are supposed to eat the oil, because you could sterilize the very thing that's going to eat all the oil up for you. So again, we don't want to mess up this soil microbial community. Yeah, and that reduces the toxicity to the plants and the animals. All right, so it turns out that when you add oil to marsh mud, it increases the rate at which that soil decomposes. The microbial community gets faster. It's probably a different community than the one that was there. There are definitely a lot of microbes that I would assume, are stressed out, toxed out. But new bacteria take over and they start eating the oil, and they also eat the peat faster than they would have eaten it without the oil. Our dispersants or fertilizers. Some people want to fertilize the oil slick so the bacteria can eat the oil faster. In the experiments that I've seen, the ones that I've done, These do not reduce this enhancement of eating the peat. How do these things affect the hydrocarbon disappearance? Well, it depends. In one experiment, it depended on which way you measured the oil. Remember I mentioned that we've got this fingerprinting thing, and they're making it better all the time. There are other ways to measure the oil too. If you use one way of measuring oil, you say, oh, the dispersant helped the oil disappear faster. But if you looked at another way of measuring the oil, you go, no, there's no effect there. In the salt marshes, it was pretty clear the both way, I can't remember right now. I want to say maybe it's just one way, maybe two ways. I have to go back and remember this paper again. But the dispersant slowed the rate at which oil disappeared over time from jars of mud we held in the laboratory. And this really, I hope this is making you think about what's the best way to measure the oil. You know, what's the best way to fingerprint this stuff? I've got citations at the bottom of here. If anyone wants to take this this PowerPoint afterwards, you can take it and you can track down the sources of my information on these slides. All right. I think the most important way to measure the oil chemically is the one that most mimics what it does to animals. There's a couple of reasons why the animals are important. Well, they're ecologically important. Many of these guys are food. Yes? Many animals are ecologically important. They're food or predators or other animals. Many people make their living catching animals. They're recreationally important. They're emotionally important. You know, looking at the birds and the oil spill definitely could get your emotions going. And probably our physiology responds more like their physiology than the plants or than the microbes. Trying to guess how oil and dispersant is going to affect you, you're probably going to want to pay attention to these fish studies that Chris talked about more so than the plant studies and the mud studies that I've been talking about. All right. It turns out that crude oil is not the worst thing out there. I've worked with diesel oil. I've also worked with fuel oil. And these refined products are more toxic than the crude oil. Crude oil is less toxic than things we make out of crude oil. Toxicity declines over time because you've got you've got bacteria out there who can eat the oil. This one's real important to me, the one I just popped up here on the screen. The toxicity of the soil and the water are unrelated. We've seen this twice now in our experiments. Chris described his aquarium to you where he's got the oil and the mud. I mean, he's got the water and he's got the mud below. You can analyze the toxicity of the water to fish, and you can use the mud to do it to worms. And you would hope that where you had your most toxic water to fish, you'd also have your most toxic mud to worms. It turns out that's not the case because the oil is going to different places. Sometimes there's more of it up in the water, and sometimes there's more of it down in the mud. You cannot predict the toxicity of a site simply from measuring the water or simply from measuring the toxicity of the sediment. You need them both. You can imagine how complicated this gets in a coastal estuary where the tides come in and out every day. The toxicity of a site is probably changing as the waters move in and out and bring in different bodies of water. Citations. All right. When we do see differences in the lab between dispersed oil and undispersed oil in toxicity, the dispersed oil has been more toxic than the undispersed oil. Chris showed you one of these experiments. You know, sometimes there was no difference in toxicity between the dispersed oil and the Corexit. But almost always the dispersed oil is more toxic than the undispersed oil. But in the experiment, you don't see a difference sometimes. When there is a difference, though, the dispersed oil is more toxic than the undispersed oil. The toxicity is prolonged by the dispersants in our laboratory studies. Chris showed you one example. I had some more graphs I took out of here to save time, but this is not a one-time observation. Seen this in multiple studies. This effect of dispersed oil being more toxic than undispersed oil is greater at lower salinity. Again, think about the very tip end of the Mississippi River That's where the stuff first, that's where oil, you can see it first come ashore. And that's probably, if there's oil coming ashore, you might have dispersed oil in the waters underneath, also possibly coming ashore there. And in a low salinity environment, being diluted with the river water. And this one's, now I'm wrapping around. Remember we talked about the chemicals. We cannot yet predict the toxicity of a sample based off the chemical analysis of the sample. I wish we could, because the chemical, as expensive and difficult as the chemical analysis are, the toxicity measurements, as you saw Chris describe in one experiment, are even more complicated. I'll show you. Here's a graph. This is from work what I did here when I was at UL University. We were at University of Southwestern Louisiana back then, USL, UL Lafayette now, with Paul Clerks here in the biology department. And what I'm showing here, two graphs. These are our best relationships between chemistry and toxicity. The lines represent a statistically significant relationship between a chemical that we measured and the toxicity. What you see on the vertical axis is the survival. So higher survival at lower concentrations, and then as the concentration picks up, you see the survival go down. So the concentration of the compounds are on the horizontal axis. But look at the spread of the real data points around those lines. If we're using the chemistry measurements to decide what is clean and what is not, where to spend our limited resources, you're going to get some of them wrong. You're going to end up spending money to clean up sites that really aren't all that toxic, even though they have a high concentration of these chemicals. And unfortunately, you're going to end up not cleaning up some sites that are actually quite toxic because they don't have a high concentration of these chemicals. I wish I knew the answer. I mean, this is the best chemistry we got, but the chemistry does not yet allow us to predict toxicity accurately. This is not just our work. Chris showed you some of that work. Another repeat study, Adam Kuhl was a senior author. There's these two previous studies. There's another one I've seen recently. This is not, we're not the first people to show this, certainly, and we're not the last people to show this, but this is one message that isn't out there. I was at a meeting a couple of years ago. Some people from Coast Guard and NOAA from Washington, D.C. were talking about how they wanted to end toxicity testing and oil spill response because it was not matching what the chemistry told them. And that is bass-ackwards. The toxicity is what is important. If it doesn't match the toxicity, then the chemistry is not right. It's not the other way around, in my opinion.
What's TTAH?
This is total target aromatic hydrocarbons. We measured hydrocarbons, I want to say 8 different ways in this study, and tried to see which was the best one, and these are the 2 of the best from that study. All right. So yeah, I believe that the best chemical measurement is the one that most closely matches the toxicity. And I trust, we have to use chemistry data, so I trust the fingerprinting over the other things that are out there, such as total petroleum hydrocarbons. There's some fluorometry measurements out there. The fingerprinting is the best we have, but don't start thinking it is representative of the toxicity. All right. I'm now going to switch gears and start talking about some of the things I've learned about dispersants and surfactants as people have contacted me since the spill, and Chris and I started working with people trying to develop a new and better dispersant. All right. So a dispersant I mentioned earlier is something that we use to try to break up the oil slick, because you want to prevent it from getting to the marshes, you know, like Emily showed you. We want to prevent that. We want that oil to be dispersed, become tiny drops, and get eaten by the bacteria far away from our coastal areas. The dispersant is made up of many chemicals itself. One of the most important types of chemicals in a dispersant is called a surfactant. This is a word someone invented, I don't know, 50 or so, maybe more or so years ago. It's a shortened abbreviation of surface active, because it goes to the surface of that oil slick and starts breaking it up. It acts on the surface and and breaks up into little tiny drops like Emily showed you. There are thousands of surfactants currently on the market. None of us buy surfactants by themselves. They're just in things that we buy. They're in our shampoos, they're in our dishwashing detergents, they're in our foods, they're everywhere. Well, there are many things. People have been making surfactants for thousands of years. I remember reading about, the Romans making soap from animal fats and ash, and they're making surfactants, and you can also do it with plant oils and ash. Starting in the late 1800s, people were figured out how to do this, make surfactants from crude oil, from petroleum. This is the building blocks. They could break the oil down, and the organic chemists, what they can do with oil is impressive. But there's a lot of surfactants now. That's what a detergent is. A soap is a natural-based surfactant. A detergent is an artificially based surfactant, generally from petroleum. Since the late 1800s, though, there's another way to make surfactants. And this is, you brew it, basically. You use bacteria to brew your surfactant. It's a lot higher tech, though, than brewing your own beer. These are designed microbes, genetically modified or engineered. They're designed so that they cannot reproduce. They're going to spend their life of a few days doing almost only one thing, that's making the chemical you want them to make. You then take that chemical and purify it. You can purify it the way an organic chemist would, or you can use there are alternative techniques to purification now called green chemistry. Here are some examples of surfactants, and if you pick up some of the foods at your house or some of the shampoos or detergents at your house, you might see sodium lauryl sulfate, you might see sodium sulfonisate, I just say DOS on it. Surfactant and FA glue are two that are microbially produced that I'll talk about a little bit as well. So here's this, here's a table. I want to talk about the kind of traditional organic chemistry where you start with petroleum, you make chemicals using catalyst, heavy metals sometimes, pressure, heat, pH, and you're going to use solvents to get your product out, and then you're going to go use that product in shampoo or in Corexit. With this new approach, you're using agricultural products. Glucose right now is the only way that they can do it financially compatible or competitively with petroleum, but they would like to move in and start doing it with agricultural waste products. It's a fermentation process, a single batch over a couple days, and you can use the same solvents as a organic chemistry, or you can use some of the newer green chemistry stuff. I got contacted after the spill by this company called Modular Genetics. I'd never heard of any of this stuff before then. They've been in business. He talked about, he's got this vision, the guy I talked to over there, Kevin Jarrell. He's got this vision that he says that one day petroleum will be too valuable to use for shampoo and detergent. One day you're going to have to use you're going to have to brew that stuff instead. You're going to brew it from either glucose, or maybe better, you're going to use bagasse from sugarcane or rice hulls, something left over that's not being used, you're going to brew it. And that's how you're going to get the fragrances and the detergents and the surfactants. Sorry, you get the fragrances and the surfactants that you're going to need to make your shampoo, for example. And so that's kind of nice. Let me know when that happens. Just know we've been in business since the 1980s. We compete against organic chemists. We sell this stuff. I can't, and he says some of our product is probably in stuff that you use. I had no idea this was going on. He says, we want to make a better surfactant. We want to design a dispersant, and we want it to be less toxic than what's on the market. I said, well, I'm kind of interested in that. Let's see if we can come up with an experiment, or something scientifically interest us. And so where we are now, EPA has put up the money for us to do some science. And so they're giving money to Iowa State University, LSU, and to Columbia University. Modular Genetics has given the microbes and the surfactants to all three universities to do the work on. Iowa State, what they're working on, they're trying to figure out how to make the fermentation more effective using agricultural waste products so that they don't have to use glucose anymore. What we're trying to do, and that's their interest, what Chris and I are trying to understand is the toxicity of oil and this Corexit and alternatives. And so we're using this stuff to compare the toxicity of the products that they're making versus the traditional surfactants. Columbia University, those guys, they study the surfactantness of a molecule, if you will. They study how well it breaks up an oil into tiny drops of water. Or you can also break up a water slick on oil into tiny droplets. I think they do both of those things over there. But those are the three groups that are working in the project that's funding us right now. All right, so let's talk about the toxicity of some of these surfactants to fish. I'm going to talk about two petroleum-based surfactants, SLS and DOS, and I'll talk about two microbial-based surfactants. Surfactin is one of the first described surfactants, I think about almost 100 years ago. It's microbially produced. It's been used as an antibiotic. It's one that evidently modulogenetics sells to people who use it in other products. They've also got this new one that they used as a base, the surfactant as a base. They said, this has got some good properties, but we couldn't use this to make a good oil spill dispersant because surfactant doesn't dissolve in water very well. So they designed some microbes to make surfactant a little bit differently and in a way that would make it more water soluble, and that's what the FA glue is. All right. The first thing I'm going to talk about is Corexit. This is a list of ingredients for Corexit. You don't see any of these surfactants in there, but because it's probably buried down here in this row right here where it says distillates petroleum hydro-treated light. That's like saying the rest, this is like I'm telling you the recipe for gumbo is get some vegetables and some meat and put it on rice. Vegetables could be anything, and that's kind of what this distillates here is. That's almost anything as far as I can tell. But we know that DOS is one of the surfactants in Corexit based on the research by that research group there. So Chris's group has done some work with the DOS, and they've looked at the toxicity as it's affected by salinity. And it's a very interesting pattern here, because what you see is as the salinity goes up, the toxicity of this stuff declines. Now, so at very low salinity, it takes only 10 parts per million to kill half the fish. But if you get the salinity up around 5, you've got to raise it to 30 parts per million to kill half the fish in 48 hours. And then it comes back down again. What we think we're seeing here is that the fish is trying to keep, the fish has a salinity inside its body. We all have a salinity inside our body. And one of the things, it's much easier for us to maintain our proper salinity than it is a fish. Because the fish has those gills and salt's always trying to get in or get out. It's got this battle trying to keep its body at the right salinity. And the salinity of the fish is up here in the middle somewhere. So as the salinity is really fresh or really salty, the fish has to work harder to maintain its salinity properly. So we think that's why we're seeing this pattern here. Very different than the pattern we saw with dispersed oil. Dispersed oil did not show this pattern. Dispersed oil is more toxic the fresher it gets. That's a little clue, one of the clues that tells me the problem with dispersed oil is not the dispersant, it's the oil part or the dispersed oil part, but it's not the dispersant alone. But there are other things in Corexit too. There could be other things in Corexit causing that pattern. This is only one ingredient within the Corexit. Now I'm going to talk to you about another surfactant, the SLS, sodium lauryl sulfate. This is a list of ingredients in Dawn dishwashing detergent. And here we compared the toxicity of the SLS, which is the triangles on here. We use the FA glue and the surfactant. So the SLS is a petroleum-based surfactant. The surfactant is a biological one, but the natural one. The FA glue is the engineered one. Notice that very different pattern with salinity here. Implies that the mode of toxicity is not related to the osmolar regulation across the gills as it was with DOS. The saltier it gets, the more sensitive these animals are to the substance. You also see that, whoop, wrong one. You also see that, there's my red. You also see that the FA glue, the designed one, did not become as toxic as salinity increased as the other two did. So we're using the salinity of the water to help us see if the reason the substance is toxic is the same. Very different. So we got two petroleum-based surfactants here, DOS and SLS, two very different patterns in the response to salinity. Two very different ways these chemicals are affecting the fish. And then also, you know, this designed one is less toxic than the natural one, and also than the SLS, and less affected by salinity. So something's going on there. Pardon me a moment. I'm from New Orleans. I got to do that. I had to. All right. And so where are we now? Where are we now? We now are at the stage where we're comparing the toxicity of different shapes of FA glue. It turns out that FA glue comes in two different shapes. If you look at your right hand and your left hand, they both do the same thing, but they're mirror images of each other. They're different shapes. These different shapes of FA glue chemically seem to act the same. They have very similar solubilities. That's one reason why it's so hard to separate them. But we're working with some really smart people at Modular Genetics in Iowa State who can separate these things. And Chris is starting experiments next week to compare the toxicity of these different shapes. And this is important because if we can understand, first of all, if there's a difference in the toxicity of the different shapes, then we can more quickly understand the relationship between the shape and the toxicity. And then other scientists can then more efficiently design less toxic molecules that are also good surfactants. And so that's where the research is going now. All right, I've got one more slide that I'm going to show. I just added this afternoon. I almost hate to show it because it's almost a distraction from the things. I've already told you everything I pretty much want you to know. Dispersed oil is more toxic than undispersed oil. It stays toxic longer than undispersed oil in the laboratory setting. That effect is greater at lower salinity. And that's just acute toxicity. We've still got work going on with the survivors of these experiments. That's the most important thing. But people continually want to know, people are worried about this dispersant. And they've had trouble trying to put a frame of reference on it. How toxic is it? And so it'd be really nice to compare the toxicity of the dispersant to things that you're familiar with. I just found this, I read this abstract just before Christmas, and this afternoon I got a copy of this reprint, and I can compare it compares the toxicity of Corexit to Dawn dishwashing detergent and Johnson's Baby shampoo. So here's that stuff. I'll read the words first and we'll walk through the diagram. Corexit is less toxic than Dawn dishwashing detergent. It's more toxic than Johnson's Baby shampoo and Greenworks all-purpose cleaner. I'll walk you through the diagram to show it to you. So here's the Corexit over here, and so here's the concentration. You got to get up to around about 50 ppm to kill half the fish. For the Dawn is the product A. You don't have to get the concentration of Dawn as high to kill half the fish as you do for the Corexit. Here's Johnson's Baby shampoo over here, product F, and here's that green detergent, green cleaner over there, Project H. When I first wanted to see this answer for myself, I thought this would be a beautiful hook to get people in and say, look, this dispersant's bad stuff, but that's not what's important. What's important is the toxicity of the oil dispersant mixture and how it degrades over time. This is a kind of a distraction from that really important question. How does the dispersed oil lose its toxicity? How fast does it lose it? What conditions causes it to lose its toxicity? But people always want to know this. I got the paper today, so I'll put it at the end of this talk. And I'll come back to that, I guess. So yeah, we'll move to the questions. If Chris will come up here, we'll take some questions now. Here are our email addresses. We also have a Facebook page where we